Another season. Another morning. Another challenge waiting to unfold. This is the amazing race of the 2024 Winter Science Factory participants. 3 contenders. 3 different missions. But time? That's the real competition. First move is made. Glances exchanged. Plans forming. No words needed. They move. Coats on. Pipettes in hand. And the race begins. Jaehee's mission commands successful isolation of mouse splenocyte T cells. Currently, she is removing the spleen from the cervical dislocation mouse. She will then use EasySep to use magnetization properties to isolate primary T cells. We asked Jaehee to explain what EasySep is.
EasySep is a immunomagnetic separation system that allows the separation of target cells using magnetic properties. We need to attach antibodies to the surface antigens of our target cells. Then we use this magnet to separate those cells so that we get the isolated cells that we targeted.
Gun has also received a mission. The task? To successfully perform cell staining and confocal imaging of Jurkat-Sinach cells. She had this to say regarding the mysterious envelope that she got.
Honestly, I was so overwhelmed, but I thought it was some kind of love letter or 용돈, but it was actually some kind of weird mission. But since I got it, I gotta do it anyways.
The mission requires precise staining of Jurkat-Sinach cells, followed by confocal imaging to visualize receptor expression. This process is crucial for their research on PDL1 targeted synthetic receptor engineering for T-cell immunotherapy in solid tumors. Let's now see how the other participant is doing with his mission. Like the others, Yaphet has also received a mission. The challenge? To label mTORCers on HEK cells, apply magnetic stimulation, and capture confocal images, to reveal CFO's expression. Here's what he shared when asked about the mission.
I've heard this experiment is like assembling IKEA furniture without instruction, but the more you get to do the experiment, the more you get into it, the more confidence you get that this thing might actually work, the more assurance that you get that in the end you might actually find what you're looking for. And I'm excited and eager to see that thing come to life.
After labeling, the cells need time to attach to the mTORCers. Only then can he move to the crucial part, which is the magnetic stimulation. So, first, she stained the cells with the appropriate antibodies. This step helps us track the expression of the synthetic receptor we engineered.
So magnetic stimulation is the heart of my experiment, and by applying force to the mTORCers, I hope to trigger a cellular response, one of which is the expression of CFOS. That is our indicator that mechanosensitive signaling has occurred.
Clock ticking, and mission? Still underway. Will Gun successfully complete the task? In the meantime, let's see what Yaphet has been up to. The final step, confocal imaging. If all goes well, Yaphet will capture fluorescent signals indicating CFOS expression. While we visited the other challengers, Yaphet had finished conducting ICC for his samples. Will he ace this challenge as the other competitors? Prove that the magnetic stimulation was successful?